Hello, my name is Ahmad Rosri from AS222AM1. Today, our group will present about sweet protection, Trehalose and future biopreservation in medicine. This video is about a very special sugar. It helps tiny animals survive freezing. It also helps plants survive drought, and it will help us save more life in medicine. You might have heard of tardigrade, also called the water bear. This tiny creature can survive in space. It also can survive in extreme cold. One reason is the sugar we call trehalose. We will look at how trehalose works. We will see why it matters now, and we will learn about new research using trehalose to help people. First thing first, I will present about the cryopreservation crisis. Every year, millions of people need medical treatment that use preserved cell stem cell therapy, organ transplants, blood storage, IVF treatment, and all of these need to freeze and keep cell alive. The problem is the chemical we use today to freeze cell have serious issues. One is called DMSO, another is glycerol. They damage cell. They also can be toxic and have a side effect. This limit their use. In fact, DMSO is so harmful that scientists must be very careful. Use too little and cell get the damage by ice. Use too much and the chemical kill the cell. It's a difficult balance. Because this chemical do not fully protect cell, many patients sample are lost. This limit what doctor can do. It limit organ transplant and limit new cell therapies. But nature already has a solution, a molecule that has been solving this problem for millions of years. Next, we move to natural solution of trehalose. It is a simple sugar found in some of tough organisms. The tardigrade or water bear can survive extreme dryness, freezing, and even the vacuum of space. Trehalose help them to do this. Desert plants that survive years without rain, they use trehalose. Bakery yeast that can be dried and brought back to life, trehalose protect it. Even some shrimp in Antarctica use trehalose to survive freezing. So, why is trehalose special? First, it is non-toxic, unlike DMSO. Second, it breaks down naturally in the body. Third, it comes from nature. Evolution has already perfected it over millions of years. Scientists call trehalose evolution perfect preservation system. When you understand how it works, you will see why. Let's take a look at what this molecule looks like. Trehalose has the chemical formula C12H22O11. Its molecular weight is 342.30 grams per mole. It is made of two glucose units connected by a special bond called the alpha-alpha-1,1 glycosidic bond. This is not normal sugar linkage. Look at the 2D structure on the left. Most sugar like sucrose are connected differently, but trehalose has a 1,1 linkage. This means the two glucose units are connected directly at both ends. This unique connection gives trehalose a rigid shape. Scientists call it a clamshell conformation. And because of this structure, trehalose is non-reducing sugar. This means it is more stable and does not break down easily. Now, let's look at the properties that make trehalose so useful. First is the glass transition temperature, or TG. This is the temperature where a substance forms a glass-like state. Trehalose has a temperature of 114 degrees Celsius. This is the highest of all sugar like it. For comparison, sucrose has a temperature of 60 degrees Celsius. Maltose has a temperature of 70 degrees. This high temperature means trehalose forms a stable glassy state at room temperature. This is how it protects and traps biological molecules. Trehalose has a multi-point of 203 degrees Celsius. Again, this is the higher than sucrose at 186 degrees. The height multi-point shows that trehalose is very stable. It can be processed without breaking down. Trehalose dissolves very well in water. When it dissolves, it does something special. It organizes the water molecule around it. Scientists call this a cosmotropic effect. Trehalose forms a water shell that is over 3 molecules thick. This organized water does two things. First, it protects cells from ice damage. Second, it shields cells from harmful chemicals that can use damage. Trehalose has a log P value of negative 3.7. This means it is highly water-loving and polar. The more negative the log P, the more substance dissolved in water. But this is also a challenge. Trehalose is so water-loving that it cannot cross the cell membrane on its own. This means we need clever ways to deliver trehalose inside cell. Finally, and most importantly, trehalose has 8 hydrogen and bond donors and 11 hydrogen bonds acceptors. That is a total of 19 sites that can form bonds with other molecules. This is the highest hash bonding capacity of any natural sugar. Sucrose has only 17 sites. Maltose has 15. Glucose has 30. This high bonding ability is exactly why trehalose is so good at protecting biological structure. What does all this mean? Trehalose can form a stable glassy protective shell around cell, protein, and membranes. The shell allows survival through freezing and drying. It is why tardigrade can survive in space. and it is why trehalose is one of the most promising molecules in medicine today.
The first mechanism is vitrification, the formation of a glassy state. On the left, you see what happens without trehalose. Sharp ice crystal form, and you can see them piercing the ceramic membrane. The red X indicate membrane punctures. The proteins inside become denatured, and the cell dies irreversible damage. Now, look at the right side with trehalose. Instead of ice, trehalose form a smooth, glassy matrix, a vitrified state that surrounds the entire cell. You can see the trehalose molecules shown in orange embedded in this protective glassy shield. The membrane stays intact, protein remain folded, and the cell survives. This is a power of vitrification. Trehalose prevents ice crystallization by forming a high viscosity glassy state that prevents cellular integrity. The second mechanism is the water replacement hypothesis. Let's look at the three-panel comparison. In the first panel, we call normal state. Water molecules stabilize the protein, keeping it folded and functional. This is the native active state of the protein. In the second panel, dehydrated or stressed. Water is removed. Without water, the protein collapses and becomes denatured. It loses its 3D structure and its function. This is what happens during freezing or drying. In the third panel, trehalose protected. Trehalose molecules step in. They form hydrogen bonds with the protein, acting as a surrogate for the missing water. The protein is restored to its native folded structure. In essence, trehalose replaces water, preserving the 3D structure of protein and preventing denaturation. Trehalose is found throughout nature, in tardigrades, resurrection plants, fungi, insects, and bacteria. But how it's made? Through the TPS or TPP pathway, also called the OTSBA pathway. The process starts with the two substrates, glucose 6-phosphate and UDP glucose. Step 1. The enzyme OTSA, trehalose 6-phosphate synthase, catalyzes a condensation reaction, joining these two molecules to form trehalose 6-phosphate. Step 2. The enzyme OTSB, trehalose 6-phosphate phosphatase, removes the phosphate group, producing pure trehalose. For the step 3, the trehalose is now ready to protect cell. Another fun fact, humans have the enzyme trehalase, which breaks trehalose into glucose. This is why oral delivery of trehalose is challenging. It gets broken down before reaching its target. Because of its unique protective properties, trehalose has found application across multiple fields. In medical preservation, trehalose is used for stem cell cryopreservation, red blood cell storage, and tissue preservation. It replaces toxic DMSO and achieves up to 90% viability in stem cell. In food and probiotic, trehalose protects beneficial bacteria during freeze drying. A 2025 studies achieved 98% of survival, a remarkable breakthrough. In therapy, trehalose shows promise for Alzheimer disease, Parkinson's disease, kidney disease, and metabolic disorder like obesity, all through its ability to induce autophagy and protect cells. In biomaterial and nanotechnology, trehalose-based hydrogel, nanoparticle, and biodegradable polymers enable targeted drug delivery and wound healing application. Trehalose is truly a versatile molecule for the future of medicine. Now, let's look at the data that make trehalose so compelling. On the left, we have the current standard, DMSO and glycerol. These are toxic cryoprotectants. They give us about 76 viability for stem cell, but they come with a serious cytotoxicity and limited application. The cells are damaged and the approach is far from ideal. On the right, we have trehalose. It's non-toxic and biocompatible. It boosts viability up to 90%, a 14% improvement with protected intact cell. This is why researchers are so excited about trehalose. However, there's a challenge. Trehalose doesn't cross cell membrane easily because it's too polar and it's hydrophilic. This brings us to the Goldilocks zone. We need an optimal concentration between 100 and 400 millimolar. Too little, and there's no protection. Too much and osmotic damage occurs. The sweet spot is critical. Researchers are solving the delivery challenge with the three main approach. First, cell-penetrating peptides, a short sequence that carry trehalose into cell. Second, nanoparticle encapsulation, packaging trehalose inside tiny delivery vehicle. Third, selective chemical modification, altering the trehalose molecule to help it cross membrane. This is exactly what primary research paper by Murray, Kilbride, and Gibson explore, finding ways to grade trehalose inside cells safely and effectively.
Assalamualaikum Doctor, my name is Muhammad Ibn Arifin and I will continue my part for the research on trehalose. Let's look at one of the most exciting recent breakthrough, probiotic preservation. This 2025 study by Wang and colleagues published in Food Bioscience focused on Dictyphane Bacillus plantarum, a beneficial bacterium used in probiotic. The results are remarkable. The control group with no trehalose showed only 17% survival after freeze drying. There's a major loss of viable bacteria. But when the bacteria were engineered to produce their own trehalose, and additional trehalose was added externally, a combination of endogenous and exogenous trehalose, survival skyrocketed to 98%. So, the key finding is clear. Engineered bacteria producing trehalose combined with added trehalose achieves 98% survival. Trehalose protects cell, membrane, and protein during the stress of freeze drying, significantly improving the viability of probiotics for food and health application. And next, trehalose protective power extends beyond preservation. It also shows remarkable potential in neuroprotection. On the left, you see a brain without Trehalose. Toxic protein aggregates accumulate. These are the hallmarks of neurodegenerative disease like Alzheimer, Parkinson's, and Huntington's. On the right, you see a brain with Trehalose. The protein aggregates have been cleared, leaving a clean, healthy brain. This is achieved through autophagy induction. Here's how Trehalose protect the brain through 4 key mechanisms. First, it induces autophagy, which is the cell recycling system. Second, it clears toxic protein aggregates like alpha-synuclein and amyloid beta. Third, it reduces neuroinflammation. And fourth, it protects neuron from degeneration. The animation shows the autophagy process in action. Protein aggregates are isolated by a phagophore, and our phagosome forms. It fuses with a lysosome, and the aggregates are degraded and recycled. This research holds promise for Alzheimer, Parkinson, and Huntington disease. Now, let's highlight our primary research paper, Murray, Kilbright, and Gibson's 2024 review in RSC Medicinal Chemistry. This comprehensive review examines Trehalose in cryopreservation, covering application, mechanism, and intracellular delivery opportunities. The key findings are significant. First, the optimal concentration of Trehalose is between 100 and 400 millimolar. This is the Goldilocks zone, which too little offers no protection and too much causes osmotic damage. Second, the paper identifies delivery challenge and solution. Trehalose doesn't cross cell membrane easily, but researchers are solving this through 3 ways: cell-penetrating peptides, encapsulation in nanoparticles, and selective hydroxyl modification. Third, the paper compares Trehalose with sucrose, and Trehalose is superior due to its higher glass transition temperature, its non-reducing nature, and its autophagy-induced ability. The figure illustrates the 3 proposed mechanisms of Trehalose in cryoprotection: water replacement, vitrification, and membrane stabilization, where Trehalose interacts with polar head group to stabilize membranes. Trehalose is also emerging as a therapeutic agent for kidney disease, specifically focal segmental glomerulosclerosis, or FSGS. This 2025 study in Frontiers in Pharmacology demonstrates Trehalose protective effects. Trehalose provides podocyte protection. Podocytes are the filtration cells of the kidney. They are like tiny cells that form the filtration barrier. Trehalose protects these cells and reduces proteinuria, the leakage of protein into urine that appears as foamy urine. Trehalose also reduces fibrosis, less scarring in kidney tissue, and improves kidney function with better filtration rates. The mechanism is autophagy in activated kidney cells. Trehalose enters the kidney cell, activates autophagy, damages proteins and organelles engulfed. They fuse with lysosomes and cellular cleanup and protection occur. Let's look at glomerulus, the filtering unit of the kidney. Before Trehalose treatment, photocytes are damaged. You can see the process effacement. This leads to proteinuria, poor filtration, inflammation, and fibrosis. However, after Trehalose treatment, the photocytes are protected. Healthy photocytes are restored, proteinuria is reduced, filtration improved, and there are less inflammation and fibrosis. So, the overall benefits are clear. Trehalose protects photocytes, preserves filtration barrier integrity, reduces proteinuria, and improves kidney function. This shows strong therapeutic potential for FSGS. Finally, Trehalose show promise in metabolic disease, specifically obesity and diabetes. This 2025 study in Frontiers in Nutrition examined the effect of injectable Trehalose in mice on a high-fat diet. The results are impressive. Weight loss with significant reduction in body weight, improved insulin sensitivity, better glucose tolerance, reduced adiposity, less fat accumulation, and also enhanced energy expenditure, increased metabolism. Looking at the data, before Trehalose treatment, mice on a high-fat diet show high body weight, high fat mass, high blood glucose, and poor insulin sensitivity. After 4 weeks of Trehalose treatment, there's a significant reduction in body weight, fat mass, blood glucose, and improved insulin sensitivity. The glucose tolerance tests show improved glucose handling with Trehalose treatment. The mechanism is Trehalose-induced autophagy in fat tissue. Trehalose enters adipose tissue, which activates autophagy. Autophagosomes form, fuse with lysosomes, and lipid droplets are degraded, reducing fat mass and improving metabolism. In summary, Trehalose activates autophagy in adipocytes, promoting lipid breakdown and improving metabolic health, reducing body weight, improving insulin sensitivity, and reducing adipose tissue. And that's all for my part. Thank you, Doctor.
Trehalose has immense potential for organ preservation, but 3 major hurdles stand as way. First, the gut breaks down, making oral delivery is ineffective. Second, it cannot cross the cell membranes on its own. And third, getting the concentration right is vital, and too little offers no protection, while too much can be toxic. And for the solution, which is how do we bypass these issues? To bypass these issues, we avoid degradation, we can use ingestible formulations or polymer encapsulation. Across the membranes, we use cell-penetrating peptides or nanoparticle delivery. And when it comes to concentration, we must target the Goldilocks zone, which is the optimal level for cell survival without toxic. Looking ahead, the future is very bright. There is a development of Trehalose hydrogels and polymers for targeted release and even using Trehalose nanoparticles for CRISPR delivery to brain. The ultimate goal is to hold organ preservation, which is keeping donor organ viable for longer to save more lives. The potential is immense. Trehalose-based solutions could revolutionize transplantation and save thousands of lives. Thank you.